Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your antimicrobial substances in a milk. Okay, so before starting the lecture, we have to know that what is this antimicrobial substances. Okay, any idea what is this antimicrobial substances? See, we all know that microorganisms are present into a food. Okay, these microorganisms will harmful the quality of the milk. Okay, we all know that microorganisms are present in a milk, and this microorganisms ultimately what they do, they spoil the milk. Okay, now the substances which is used to kill this microorganisms, this is called what? This is called a antimicrobial substances. Okay, or which prevent the growth of microorganism. It is also called a antimicrobial substances. Okay, now today we are going to discuss the substances, antimicrobial substances which are present into a milk. The next one is, now see what microbes do with the milk. Okay, the first one is your germicidal period, that is few minutes to few hours. Okay, this germicidal period have occur few minutes to a few hours, like your lysozyme, lactoperoxidase, thiocyanate, and other etc. Okay, the next period is your soaring period. It is several hours to several days. Okay, this soaring period is occur between a several hours to several day, and it is done by a acid producing bacteria. Okay, then the next period is your neutralization period. It occurs between a several days to a several weeks, and it is done by a yeast and mold. The next period is your putrefaction period. It occur until being a clear liquid. Okay, and it is occur by microorganisms, that is your dormant bacteria, yeast, and mold. Okay, now the next one is your germicidal properties of milk known for many years. Milk from a healthy animal contain different level of inhibitory substances. Okay, if we see a healthy animal, then the healthy animal's milk contain a different level of this inhibitory substances. Okay, this restrict the normal development of certain bacteria and sometimes even kill them. This inhibitory substance, what they do, either they restrict the growth of the bacteria, or either they destroy the bacteria. The degree of inhibitory activity varies considerably in milk of different species. Maximum activity present in a human milk. Okay, see, these inhibitory activities are maximum present in a human milk. That's why it is called that the human milk are good for the babies. Good for the babies. Okay, that's why we call it. Now the next one is there are two groups of antimicrobial substances. One is your inherited or naturally present in a milk. Okay, and the another one is your non-inherited compound, which gain entry into a milk from other sources. Okay, first of all, this antimicrobial substance are what? Which inhibit the growth of the microorganism. Inhibit the growth of the microorganisms. Okay, so we inhibit the growth of the microorganisms either inherited or either non-inherited. Okay, this inherited are those type of substances which are naturally present in a milk, and what they do, they inhibit the growth of the microorganisms or kill the microorganisms. Okay, now the second one is your non-inherited compound, which are not present in naturally in a food, but it enter into a milk from the other sources, and with the help of this, they will what they do, they will inhibit the growth of the microorganisms, or they will kill the microorganisms. So here we are going to discuss one by one, that is your inherited substances and non-inherited substances. Now the next one is your natural occurring antimicrobial substances. Okay, so first of all we are going to discuss the natural occurring antimicrobial substances. They are what, sometimes they are specific and sometimes they are non-specific in conferring protection to the newborn. Okay, if we talk about the specific, then they produce a specific type of antigen. Okay, like your immunoglobulins, complement, macrophages, and bifidus factor. Okay, now this non-specific factors produce less specific antimicrobial activities, like your iron binding protein, like lactoferrin, lysozyme, lactinins, lactoperoxidase, and vitamin binding protein and fatty acid. So here what should we do? We are going to discuss one by one. The first we are going to discuss about the specific factors, which is, that is your immunoglobulins and complement and macrophages and bifidus factor. Then after that we are going to discuss the non-specific factor. Okay, now the next one is, okay, now the non-inherent antimicrobial substances which are not naturally present into milk, but with the help of other sources it will enter into the milk and perform the activities, like your antibodies, antibiotics, sanitizers, and other chemical substances. What they do, they inhibit the growth, they kill the microorganism, or either they inhibit the growth of the microorganisms. Okay, they will use in a dairy industry and gain access to milk and exert antimicrobial activities with the different properties, with the help of different properties. Okay, now the next one is your specific factor. Okay, so here first of all we are going to discuss about the um, inherited substances or natural substances or natural substances. Okay, now the first one is your specific factor, that is your immunoglobulins, complements, macrophages, and bifidus factor. Okay, now the first one is your immunoglobulin, that is called Ig. Okay, these factors are responsible for the immunological protection of the newborns. Okay, these immunoglobulins are responsible for the immunological protection of the newborn. Newborns, why we said that newborn? Because their immune system are weak. Okay, that's why we uh, this immunological globulin protect the immunological protections of the newborns. In human, parental immunity is confirmed primarily through the transmission of immunoglobulin from mother across the placenta to the fetal circulation. Circulation. Okay, if we talk about the human, then the parental immunity means that the immunoglobulin are transferred from the mother to the fetal with the help of the placenta. Okay, so this is called what? This is called a parental immunity, means that the fetal gain the immunity from the parents. And how they gain? They gain when the immunity, this immunoglobulin are transferred from the mother to the fetus, and it is transferred with the help of placenta. Okay, now the next one is your buffalo and cow rely on the transfer of immunoglobulin from the colostrum into a newborn circulation postnatally. When gastrointestinal tract is permeable to intact a protein molecule. Okay, now IgG is the chief immunoglobulin of milk. Okay, if we talk about the type of this immunoglobulin, then this Ig, capital G, is the main immunoglobulin present into the milk, and it is confirmed as a passive immunity in a cow and buffalo. Okay, they are considered as a passive immunity. Now the second immunoglobulin is IgA, and it is considered as a passive immunity in human being, except ruminant lactal secretion. Now this IgA is dominating Ig in all external secretion of the body. Okay, suppose that in a human being, if the main secretion is Ig, then it is called a secretory Ig capital A. Okay, and the concentration of IgA in a colostrum up to about 50 milligram per ml, which drop to 1 milligram in mature milk. Okay, now the next one is your what is the function of this IgA? Okay, so first of all, it neutralizes the toxin and viruses. Then it also inhibit the bacterial adhesion and prevent the absorption of the food antigens, and also suppress the growth of the bacteria, aids in phagocytosis by bringing about the attachment of the bacteria to the macrophages. Okay, if we talk about the human milk, then it will contain a anti-Escherichia coli IgA. Okay, it is present where it is present a human milk, and it is identified as a active against a infantile diarrhea. Okay, now in colostrum IgS, which make about 70 to 80% of the total protein contained, whereas in mature milk, immunoglobulins account for only 1 to 2% of the protein. Okay, now the next one is your the complement system also play a major role in the host defense mechanism. Okay, this complement systems also play a major role in a host defense mechanism, which have a which have against infectious microbes. Okay, and it is complex systems of over 20 different types of protein which can be activated with the help of antigen and antibody complexes. Okay, this protein are activated by our antigen antibody complexes, by a certain type of carbohydrate, or by a variety of substances that are not protected by our natural inhibitors. Okay, now if we this killing of the microorganisms, clearing of the immune complexes and enhancement of the antibodies response, they are called a major biological function of the complement. Okay, so see, these three are considered as a major biological function of the complement. What? The killing of the microorganism, the clearing of the immune complexes, and the enhancement of the antibody. Okay, so there are 9 components of the complement have been detected in human being, but these 9 components are what they are present in very small amount, and they are also associated with the bactericidal properties of the milk. These also add to the defense mechanisms due to the lysis of variety of potentially pathogens. Okay, now the next natural substances present in a milk are macrophages. Okay, now viable leukocytes are present, and these leukocytes are present at a different concentration, that is your 2 into 10 to the power 6 cell per ml in a human clostrums, and it is decreases 2% of this value in a normal milk. Now, if we talk about colostrum macrophages that are associated with the phagocytus, then their percentage is about 80%. Okay. Example, engulfing and subsequent killing of bacteria, many of which are pathogenic in nature. Okay. Now suppose this is your diagram. See here the receptor, either this is your phagocyte, this is your lysozyme, and this is your soluble detector. This is your phagocytus cycle. Okay, now the next one, this diagram is the stages of the phagocyte, which include the engulfment of the pathogen and the formation of the phagosome, the digestion of the phagogenic particle in the phagolysome, and the explosion of the undigested material from the cell. See, this is your bacterium. This portion is called a bacterium, and then this portion is called a, just a minute, phagosome. Then it is undigested waste. This is your lysozyme, contain a digestive enzyme. This is your phagolysosome. Okay, and this is your macrophage. So this is the diagram for this. Now the next factor is your bifidus factor, and this bifidus factor is what is a compound that especially enhance the growth of the bifidobacteria in either a product or in the intestine of the human. Okay, this bifidobacter, what they do, they increase the number of the bifidobacter. Okay, and their number either increase in the product or either it is increase in the intestine of the humans. Okay, then human milk contain this bifidobacters, and it is responsible for the dominance of the bacterium called Bifidobacterium bifidus in infant. Okay, so in a market there are several products who have bifidogenic factors. Okay, and such several prebiotics and methyl and acetyl D-glucosamine in human milk. This bifidobacter also support the growth of the lactobacillus, and it is beneficial bacteria which protect the human body from a different type of bacteria, and the bacteria are also harmful, and how they protect it by creating the acidic environment, and in an acidic environment these bacteria cannot be grow. Okay, now this bifidobacteria are one of the major genera of bacteria that make up the gastrointestinal tract microbiota in mammals, and some bifidobacter are used as a probiotics. Okay, before in 1960, bifidobacter species were collectively referred to as a lactobacillus bifidus. Okay, now the next one is beneficial effect. In breastfeed infant, mother's milk having a bifidus factor, and this bifidus factor help in the growth of the bifidobacterium. This is in turn has this beneficial effect. Inhibition, support the growth, acidic pH in stool and synthesis. Okay, now the first one is your inhibition of the enteropathogenic E. coli, Shigella and other pathogens due to the production of the lactic and acetic acid. Okay, so due to the production of this lactic acid and acetic acid, this bifidus factor factor inhibit the enteropathogenic microorganism, that is your E. coli and Shigella. Support the growth of bifidobacterium species like your lactobacillus species, and other probiotic is a beneficial bacteria that protect baby against a harmful bacteria by creating the acidic environment. The next is your acidic pH in stools that is necessary for the absorption of the calcium, iron and phosphorus. Okay, for the absorption of this calcium, phosphorus and iron, acidic pH is required, and it is maintained by a bifidus factor. It is this bifidus factor also help in the synthesis of the vitamin B complex, and this vitamin B complex help in the infant growth. Now the next is that in infant receiving baby's food, the following adverse effect can be result. The first one is your overgrowth of the E. coli in intestine, increase the alkaline pH of stool due to the growth of the E. coli, decrease in the synthesis of vitamin B12. In such cases, the bacteroid conjugate with the bile salt in intestine and reduce their function in fat absorption. Okay, bifidobacters like lactose account for 25% and total dietary CHO. Due to the large amount of the lactose in the mother milk, not all is digested. A small portion is left and fermented in small intestine to a lactic acid. Lactic acid increase the acidity in the lower intestine, which promote the growth of the microorganism. This lactobacillus bifidus. This lactobacillus bifidus also help in the prevention of the growth of the other undesirable microorganisms, and this lactobacillus bifidus is also found in a breastfeed infant. Okay, the infant who are in breastfeeding, um, in their body also this lactobacillus bifidus are present. Non-specific factors. Okay, so the specific factors are completed. Now we are going to discuss the non-specific factor. The first one is your iron binding protein like lactoferrin, then lysozyme, then lactoperoxidase, vitamin binding protein and fatty acid, and last one is your lactanins. Okay, now lactoferrin, that is iron binding protein. Lactoferrin are also known as a lactotransferrin, is one of the important antimicrobial substances present in a milk. Okay, uh, it is a red glycoprotein that resembles blood serum transferring and is found in varying concentration in milk of different species. This lactoferrin are what? It is a red glycoprotein and it is very resemble to the blood serum transferring. And it is found in a varying concentration in a milk of different species. Human milk has a higher concentration of this protein. Okay, this lactoferrin protein or iron binding protein, concentration of this are higher in a human milk as compared to the cow or a buffalo. Okay, if we talk about the concentration of this lactoferrin in a colostrum of the human, then it will contain around 1.2 gram per 100 ml, and in buffalo it is 100 milligram per 100 ml falls during our lactation. See, the difference is very large. In human it is found around 1.2 gram per 100 ml, but in buffalo it is found around 100 milligram per 100 ml. Okay, 1 to 4% of the lactoferrin in human milk is saturated with the iron. Okay, now it is mainly a whey protein, like 70% of the this iron binding protein is whey protein, and the 30% is are casein and fat. The inhibitory effect of lactoferrin is totally based upon the ability of the squeeze out iron from the medium, okay, thereby making it unavailable to the bacteria essential for the growth. Okay, if we require some bacteria that will grow into the milk, then what should we do? We unavailable this protein, because this protein are squeeze out iron from the medium. It has antibacterial properties against some microorganisms, and the species of that microorganisms are Staphylococcus aureus, Staphylococcus albus, Pseudomonas aeruginosa, Vibrio cholera, Clostridium tyrobutiricum, Bacillus stereothermophilus, and Bacillus subtilis. Okay, this uh, apolactoferrin, that is also called a iron-free lactoferrin. It is present around 0.2 milligram per ml concentration in milk, inhibit the growth of the microorganism, that is your Enterobacter aerogenes and E. coli. Okay, now the next is your lysosome. Lysosomes are present in a different quantity in a milk of different species. Okay, suppose that in a cow milk it is present in a different amount, in a human milk it is present in a different amount. So this lysosomes are totally depend upon the types of the species. Okay, according to them, the concentration of this lysosomes are present in the milk. If we talk about the human milk, then the average lysosome content is around 30 milligram per 100 ml, is 3000 times more than that of bovine milk. Okay, such significant difference in the lysosome that contain have a physiological or nutritional implication on infant feeding. Now, breastfeed infant have a greater resistance to entering infections than those feed on artificial foods. And lysosomes are possibly associated with the mechanism of the natural immunity and local resistance. Most of the gram-positive bacteria are consider as a very sensitive to the lactic action of this lysosome due to the presence of the hydrolysis of this beta 1,4 glycosidic linkage of the peptidoglycan component. Okay, however, the gram negative bacteria are normally this lysosome resistance. Now this is your gram negative and gram positive. Okay, this this is your portion. Okay, this is considered as a protein. Okay, and this green line is considered as a liposaccharide. Okay, this membrane is called outer membrane. Then this is called a lipoprotein. Then this purple diagram is considered as a peptidoglycan. Then this will consider as a periplasmic space, and this is what considered as a cytoplasmic membrane, and this will consider as a porine. This will consider as a lipopolysaccharide, lipopolysaccharide, and this will consider as a protein. Okay, so this is the diagrammatic description of the gram negative bacteria, and gram positive bacteria. Due to the presence of this peptidoglycan, the name is suggested that as a gram negative and gram positive. Now the next one is your mechanism of this lysosome. Lysosome digests the bacterial cell wall by breaking beta 1,4 glycosidic bond, and this beta 1,4 glycosidic bonds are present in between the N-acetylmuramic acid and N-acetyl glucosamine. Okay, this beta 1,4 glycosidic bond are present in between the NAM and NAG. Okay, and what lysosome do? Lysosome digest the cell wall. Okay, now the next one, again this is a diagram of the bacterial cell wall of the gram positive bacteria and bacterial cell wall of the gram negative bacteria. This is the peptidoglycan chain which is formed by a alternative subunit of N-acetyl glucosamine and N-acetyl muramic acid linked by a beta 1,4 glycosidic bond. Okay, this is your peptidoglycan. This is this layer is called what? Peptidoglycan, and this layer inner layer is called a cytoplasmic membrane. Here the peptidoglycan membrane are ruptured, and when it is ruptured, then water flow come inside. Okay, and this is your infant of the water, and this is the lysis. Okay, cleavage of the peptidoglycan by a lysosome action. Okay, this diagram is what? Cleavage of the peptidoglycan by a lysosome action. Now, vitamin binding protein. Okay, we all know that there are a certain number of proteins are present in a milk, and it depend upon the species. Okay, and it is also considered as a the species of the protein that bind with the some essential vitamin family, and thereby bacteria starve. Okay, so the protein particularly bind with the two types of vitamins, that is your vitamins B12 and a folate in a milk. Okay, if we talk about milk of cow and a human, then the binding capacity of this folate protein is far excess of natural folate content. Okay, same as for a vitamin B12 in a human milk, but in cow milk there is a little or no excess of this type of unsaturation binding. Okay, so in presence of this unsaturated binding protein, the growth of the microorganism required B12 and folate is inhibited. Now see what is written in a slide. There are a certain protein present in a milk, some species that bind with some essential vitamins formally, thereby making bacteria starve that are fastidious for such vitamins. These vitamins particularly bind with the vitamin B12 and folate in a milk. In a milk of both, that is your cow and milk, the binding capacity of the folate binding protein is far in excess of natural folate content. The same is true for a vitamin B12 in a human milk, but in cow milk there is a little or no excess of unsaturated binding protein. Hence, in the presence of this unsaturated binding protein, the growth of the microbes requiring vitamin B12 and the folate is inhibiting. Okay, now the next come to the next topic, that is your vitamin binding protein have a following function, like it will regulate the bacterial growth in intestinal tract, aid in the absorption, conserve vitamin B12, and it is also play a role in mammary gland prior to a milk secretion. Okay, now first one is that regulate the bacterial growth in a gastrointestinal tract of suckling animals by denying the vitamins to a specific bacteria. Add in the absorption of the vitamin in the gut and conserve vitamin B12 by adding the reabsorption of vitamin B12 loss in a bile. Okay, now the next one is your fatty acid. Okay, milk, different species of the milk contain different type of free fatty acid. Okay, and generally medium chain fatty acid, especially like your octotonic deconoic acid and some polysaturated chain fatty acid are more active against a gram positive bacteria. Okay, if the fatty acid concentration is lower after being free from our glyceride of the milk fat are inhibitory to many bacteria like lactobacilli and lactic streptococci. Okay, this free fatty acid in milk result either from the action of the lipase naturally present in a milk or from the lipolytic activity of certain microbes. Although many organisms required fatty acid as a nutrient for the growth, but in a certain concentration. If the concentration is exceed certain limit in medium growth is strongly inhibited. Now the next one is your lactanins. The concentration of the lactanin fraction with lactin L1 and lactin L2 in milk varies. Okay, if we talk about the lactin, then the fraction with and the concentration of the lactin fraction is lactin 1 and lactin 2. The bovine colostrum are rich in L1 and normal human milk contain higher the quantities of L2. Okay, and the third fraction are also considered as a L3. Okay, and it is what it is also being detected in a raw milk. This L3 are detected in a raw milk. This differ from L1 and L2 on the basis of its source and inhibitory action. There is a partial inactivation of L1 at 60 degrees Celsius for 30 minutes on heating raw milk to a 70 degrees Celsius per 20 minute, and the pH will be 7.0. L1 is completely destroyed, leaving behind L2 fraction in relatively higher concentration. And it proved that there is a two compound N1 and L2 will interact as some bactericidal than either one alone. Lactanin is mostly active against a microorganism, that is your Streptococcus pyogenes. Okay, the ability of the microorganism to grow a udder is due to the fact that the lactanin has a little activity under a anaerobic condition. Additional glutathione, cysteine, and other reducing substance in milk reduces when this antimicrobial action of lactanin occur in a milk. Okay, if the lactanin antimicrobial action occur in a milk, then the glutathione, cysteine, and other reducing substances concentration will also reduces. However, other factor in the milk are stimulatory for the growth of the streptococcus pyogenes in udder that causes a mastitis. Okay, now the next one is your lactoperoxidase. It is also present in a cow milk, and the amount of this lactoperoxide in a cow milk is around 30 milligram per ml, and it is absent in a human milk. Okay, this is an enzyme which is very identical to the L2 fraction of the lactanin. If we talk about the bovine raw milk, it inhibits the action of this enzyme due to the formation of an antimicrobial system or LP system under a normal condition, and the secretion of this thiocyanine component component in a milk is governed by a nutrition of the animal. Hydrogen Hydrogen peroxide can also be contributed either by polymorphonuclear leukocyte or by a hydrogen peroxide producing the microflora, that is your streptococci. Okay, now the next is your the lactoperoxidase system is a bacteriostatic to a microorganism. If we talk about this lactoperoxidase system, then it is what? It is a bacteriostatic to a microorganism. It protects calf against interim problem and may have some role in providing the defense. The mechanism is totally to a mammary gland against a action infections. Lately, attempts have been made to exploit the LP system as a method of preservation of raw milk. This LP system are what are considered as a preservation method for a raw milk, and it is also considered as a cold sterilization. Unlike in conventional method of preservation, that is your heat treatment, there is no inactivation of antimicrobial substances and other heat label constitute of the raw milk. In these efforts, two compound, that is LP system wise thiocyanide and hydrogen peroxide are added from outside to attain their balance proportion. This is a table for lactoperoxidase activities and thiocyanide concentration in different types of milk. The first one is your type of milk. This column is for a type of milk. This is your lactoperoxidase. This is your thiocyanines. Okay. Now in cow milk, lactoperoxide is 1.4 unit per ml. In EWE, it is around 0.14 to 2.38. In goat, it is around 1.3 B. So this is in human, it is around 0.06. Okay, so thiocyanide concentration in human is around 2.6. Okay, and limit for bacterial day activities is 0.02, and thiocyanide is about 5 ppm. So this is table which describe the lactoperoxidase activities of thiocyanide concentration in different type of the milk. Now the next table is for the effect of the lactoperoxidase system on the activity of lactic starter culture. The first one is your lactic acid bacteria. It will reduce the rate of the acid production. Thermophilic starter delay in coagulation time by 4.5 hours when inculated 2 hours after LP activity. Yogurt culture, it reduce the rate of acid production and delay in coloring time by 1.5 hours. Okay, so this table is about it. Okay, this table is for a effect of the lactoperoxidase system on the manufacture of the fermented milk, like dahi type of the milk, buffalo. It will lower the deacetyl and acetyl content and lower the proteolytic activities. Okay, now the next one is your significance of the antimicrobial substances. The first one is your health benefit. The primary significance of the antimicrobial substance that it will detect in milk can be related to their possible role in conferring protection to mammary gland and in neonates in mammals. The newborn therefore should be essentially be given the colostrum, which is very rich in most of these antimicrobial factor. Breastfeeding is the best for the infant. We all know that. We hear this slogan. There is lots of time that breastfeeding is the best for the infant because it derives its substances from the abundance of the antimicrobial factor in mother milk as against a animal milk based infant formula. Preservation of the milk. Although the presence of this antimicrobial factor cannot be solely to be depend upon the prevention or even to retard the significantly development of the mixed population of the microbes in raw materials, raw milk and some microbe get market affected. However, an artificial explosion of this milk naturally antimicrobial system called a lactoperoxidase system or for developing a cold sterilization like process can prove effective in preservation of raw milk. Such process hold great promise under a tropical form additions in countries like Bangladesh. Processing problems. Okay, when raw milk is used for product making, like your cheese making, then acid development in cheese vat may be retarded by inhibition of the started activities. It is due to the presence of microbial substances. Okay, if we talk about the starter culture propagation, then the heating of milk about 80 degrees Celsius is essential for the inactivation of the factors that will interfere with the normal starter growth. So this is all about the antimicrobial activity of the milk. If you have any query related to this topic, you can ask me in a comment box, or further detail you can follow the ITLS Academy on different platform like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your account, that is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe.